Hi everybody, thank you for coming to this presentation today. My name is Juliette Pearce. I'm a postdoc in the lab of Rebecca Schneider at the RWTH Uniclinic in Aachen, and I'm here to talk to you today about how we've used multi-omics data to look at the function of casein kinase 1A1 in del(5q) MDS. So MDS stands for myelodysplastic syndrome, which is a rare blood disorder that's caused by multiple different factors. So it's a heterogeneous disease, but all of these changes result in ineffective hematopoiesis, which results in the abnormal development and maturation of blood cells in the bone marrow. This disease is interesting from a clinical perspective because it has the potential to progress to acute myeloid leukemia, which depends on many factors, a lot of which are poorly understood. And this disease is commonly associated with cytogenetic abnormalities such as chromosomal translocations, insertions, and deletions. One such cytogenetic abnormality is the deletion in the chromosome 5q, so the long arm of chromosome 5. And this abnormality often indicates the type of MDS that can be treated with lenalidomide quite effectively. These patients have a very distinct clinical phenotype involving macrocytic anemia, high platelet counts, and hyperlobulated megakaryocytes. But naturally, a deletion in a chromosome like this leads to a haploinsufficiency of a set of genes. And one of these genes is casein kinase 1A1, which is one of the genes that we have decided to focus on. So what is casein kinase 1A1? It's obviously a kinase, it phosphorylates a set of target proteins, but in addition to this, it's a known signaling component of the Wnt pathway and a known regulator of p53 where it phosphorylates it to target it for degradation. One thing that we know about casein kinase 1A1 is that the haplozygosity, such as we see in this del(5q) MDS, leads to clonal expansion, but previous studies by our lab have also shown that homozygous deletion of this gene leads to bone marrow failure, so cell death. In addition to this haplozygosity, we also see that casein kinase 1A1 is frequently mutated, most commonly at position E98. And this leads to increased beta-catenin levels and cell expansion in patients. And the impact of these mutations is that these patients suffer from therapy resistance and have a much worse prognosis as a result. And we are really interested in how this haplozygosity and mutation of casein kinase interact in disease and whether we can target this to improve treatments. So how did we approach this problem? Well, we generated several cell lines, including cell lines with a wild-type casein kinase E98V, which is one of the most common mutations in del(5q) MDS, and also casein kinase haplozygous cell lines. And we collected bulk RNA sequencing along with proteomics and phosphoproteomics from all of these conditions to be able to investigate how the haplozygosity and mutation are interacting to lead to this worse prognosis in disease. So to describe the results that we have so far, starting with the transcriptional regulation, we see a good correlation between the RNA and protein levels within our samples, which is really encouraging because it's often the case that we cannot assume that RNA and protein levels correlate in this way. And when we do pathway analysis, we see several pathways which are upregulated with the E98V mutation, and these include lipid metabolism, cell death and differentiation, and protein secretion, which is really interesting to see in line with previous results that we had from preliminary analysis of this data. And in downregulated pathways, we see RNA splicing and MYC targets. And these results together suggest that casein kinase E98V, so with the mutation, has global effects on RNA and protein processing and metabolism. And these lipid metabolism and splicing related associations may be involved in ferroptosis, which is a specific type of cell death involving iron and lipid metabolism that is already known to be associated with some forms of hematopoietic malignancies. On the post-translational side, so when we look at the phosphosites that were detected by our sequencing, we see that there are many differentially regulated phosphosites. And when we do enrichment analysis on these differential phosphosites, we see changes which are very much in line with that we saw when we were looking for transcriptional regulation in these cells, including the spliceosome, again, cell cycle, and other metabolic pathways. So this is really interesting to see that we might have some patterns which are reoccurring across all levels of the regulation on these samples, which might provide us with some interesting targets. In addition to this, since casein kinase is a kinase, we are already aware of some of the targets that it would phosphorylate. So we can look at the phosphorylation levels of those sites in our data, and we do see a significant number of sites which show differential phosphorylation with the E98V mutation, and most of these phosphosites are downregulated, which would suggest that the casein kinase E98V results in a slight loss of function of the kinase. So to conclude, casein kinase 1A1 deregulation in del(5q) induces many transcriptional and translational changes that we're able to measure in this data. And this includes deregulation of RNA splicing and deregulation of lipid and iron metabolic processes, which may be really key in the process of clonal expansion and also treatment resistance in del(5q). These factors suggest an impact of casein kinase 1A1 on baseline RNA and protein processing, which is interesting because we did not do any perturbation experiments yet, so this is really just looking at the fundamental effects of casein kinase on the cells. And this paves the way for validation experiments that will hopefully reveal specific targets that may be valuable in the treatment of this disease. So to finish up, I'd like to thank all of our collaborators from Heidelberg University and the Novo Nordisk Center for Protein Research in Copenhagen, and all of the members of the lab who have worked on this. Thank you.